Okay, good morning, awesome AP Bio 2 students. It's Ms. Lynham again, ready to do another recorded lecture for you to keep you up on your AP Bio 2 class. We left off on Friday talking about operons, but some of you were kind of sleeping during class, so I wanted to go back over, start right here on the slide with the operon model, and then go over both the lac and the trip operon because I want to make sure that you guys understand what's going on. Like I said, because some of you were a little tired on Friday. All right, so the operon is, okay, here's everything that is included in the operon. The promoter, the operator, plus all the genes that are required for a metabolic pathway. Okay, the operon acts as a single transcription unit, and remember we are talking about prokaryotic organisms. Okay, talking about bacteria. Let me see if I can get my pen working today. I'm not quite sure what that's doing. All right, so in this diagram down at the bottom, we had talked about that somewhere in the prokaryotic DNA, there is a regulatory gene. This regulatory gene is in control and is responsible for the production of a repressor. Okay, and this could be next to the sequence of the genes that want to be transcribed in this case, or this could be upstream. It doesn't have to be exactly directly next to the area that needs to be transcribed. So in this case, it is kind of next to it. But here's the regulatory gene, and it's responsible for making a segment of mRNA, which in turn makes a repressor. Okay, this pressor is what can come up to the operator. Okay, here's the operator, that yellow piece, which is like the on-off switch. Okay, so that operator right there can be turned on or off. Now, what do we mean by turned on or off? If it is turned on, okay, then this thing right here is RNA polymerase. And if this is turned on, meaning this repressor in this case is not here, then RNA polymerase can come up, go like this, bind to the mRNA, and transcription will occur. So all of the genes that are down here, this might be gene 1, and you might also have a gene 2, you might have a gene 3, because in a lot of cases, prokaryotes have multiple genes that code for something that they're trying to make. In this case, it might be making lactase. So if the operator is on, okay, then this RNA polymerase can come down and transcription will occur. So this operator is a really important aspect, this on-off switch, which is located close to or within the promoter region. Okay, so we've got the promoter as well as the attachment site where you also need some the RNA poly to come and read this. So the job of the operator is to control whether or not RNA polymerase can actually bind to the promoter region. Therefore, ultimately, it's determining whether transcription is going to occur or not. Okay, so that's just kind of a little bit of the overview. Okay, so then you can see on this slide that the operon can be turned off by a protein called the repressor, like we said, that is made by the regulatory gene, which is right here. Okay, now one thing that I want you to notice is the regulatory gene is expressed continuously, although most of the time it's only making the repressor in a small supply. Okay, so if it needs to make more, it can, but this gene is always being expressed. This one is not like every other gene where it needs an on-off switch like what we're doing down here. So this one is expressed continuously. Okay, so the two types of operons, we've got the inducible operons, okay, which is the lac. Put that with that name right there. Okay, you need to associate the lac operon is inducible. What does inducible mean? They're usually in the off position. That is, they're not usually being transcribed. The reason being in this case is the lac operon is making the enzyme lactase. Well, there's usually not a reason for bacteria to make lactase because that's not their preferable sugar. They do not like to break down lactose. Their preferable sugar is glucose. And so most of the time, this gene is turned off, but it can be turned on. So it can be stimulated when a specific molecule interacts with the regulatory protein. So in this case, I want you to look at this picture. For the lac, go ahead and put your name up here. For the lac operon, the repressor in the active form, okay, it actually has these two little teeth on it right here, okay, which means that they fit into the operator. So in a normal situation, the active repressor is turning the gene off, okay, which is, if you go back up here, it says these are usually off. So the active repressor actively binds to the operator, turning transcription of this gene off. Okay, so you can see the RNA polymerase hanging down here, not able to attach to the promoter region, can't in turn go through transcription. So this is required for the breakdown of lactose. Okay, in order for bacteria to break down the lactose, this gene must be turned on. So in order to do that, it takes a little bit of a difference. So let's go through and show you the next one. Okay, so when lactose is available, meaning let's say it's in high supply, you drink a lot of milk, drink a big glass of milk, and it's available to the cytosol of the bacteria, they are going to need to break it down. So in this picture, you can see this green little molecule right here is a type of inducer that when it attaches to the active repressor, it makes it inactive. Okay, so it turns this to an inactive form. The inactivity is a result of what we call an allosteric change. Anytime you bind something not to the active site, but somewhere different on that enzyme, it can change the shape of the active site. And as you can see here, these teeth are now spread apart. There's a big gap in here, so those don't fit up into the operator. So in this case, that would turn this off. That would not be able to bind, and now we turn this whole transcription, we turn that on. Okay, so we in turn are now going to transcribe the LAC-Z, the LAC-Y, the LAC-A genes, which code for these enzymes collectively are responsible for breaking down lactose. Okay, so it's a couple of genes. There's 3 genes involved in bacteria making breaking down lactose. But these 3 genes we commonly refer to them. We just usually say lactase is the enzyme. But when you look at it more specifically, it's a couple of enzymes that work together to break down lactose. Okay, so going back to the little bullet points over here, when lactose is available, lactose binds with the repressor. Now it says lactose itself. If you study this more in depth, it's actually called allolactose. It's an isomer form of lactose. So if there's a lot of lactose in the environment, okay, in the cytosol, there's a isomer form called allolactose, which is the inducer itself, binds to the allosteric site, opposite of the active site, changes the shape now of the active site, which now it can't fit into the operator, so the transcription is turned on because now RNA polymerase can attach. Okay, so in this case, we just induced it. We turned the genes on, so now it's able to be read. Okay, so the opposite are these repressible operons. Okay, repressible is where transcription is normally on, but it can be inhibited or turned off whenever a specific molecule binds allosterically to the regulatory protein. Okay, so look at this is the form that it normally exists in. Looks like the picture we just did. So it's normally like this, which means it's inactive. It cannot fit up into the operator. So this gene, the TRP gene, this is normally on, transcription's occurring, and they're going to be making the enzymes, making tryptophan in this case, so it's normally turned on. So the TRP operon stands for tryptophan. Okay, this operon controls the production of 5 enzymes required to actually make tryptophan. Okay, so in this picture you see the TRPE, TRPD, TRPC, B, A. Those are the 5 genes necessary to make the mRNA, and this is a couple pieces like this, to make the mRNA needed to synthesize or make the ultimate product is tryptophan that's trying to be made here. So like I said, it's normally on, operon is normally on, but it can be inactivated with the presence of tryptophan, like I'm going to show you in the next picture. So in this picture, if you look up here, there's RNA polymerase attaching to the promoter region. The operator is turned on. It'll continue to go. We will get tryptophan as a result. Because why? Our repressor is inactive in its normal form. It makes an inactive repressor. Okay, how can we change that? In the next picture, you can see when tryptophan is present. When would this be? You ate a big, huge turkey meal. Okay, so because these E. coli live in your gut, so you put a lot of turkey in your stomach, tryptophan is highly available. There's no need for these bacteria to make tryptophan because that would be wasteful for them to make it and to also have it in their environment. So in this case, here's the tryptophan itself. Okay, that's you eating a lot of tryptophan and it being available to the bacteria. It acts as a co-repressor because it can bind to this section of the repressor, and by doing that, change the shape of the active site. So now look at these beautiful little teeth that can come up, fit into the operator, turn the operator off. Now RNA poly is stuck like that. It cannot go and read this, and so it's going to turn off transcription of tryptophan. Okay, once again, what caused that is the presence of tryptophan in the environment. The more of this you have, it can bind allosterically to a region of the repressor, act as a co-repressor, and now this changes the shape of the active site, and the repressor is active and can go up and bind and turn that off. Okay? So two different methods of control. So in this picture, what you see, let me draw on here for you. What you see is this one is going to turn the gene on. So this is the inducer, because here's the lactose, changes the shape, turns the gene on. Okay, this one would be turning off the repressor. So tryptophan turns gene off. Okay, so this is what is normal for the trip operon. So this one's trip, this one's lac. This is what the normal repressor looks like, but when tryptophan is in high concentration, it binds. Now this repressor fits and turns the gene off. Okay, on this one, the lac, this is the one where it turns the gene on. Okay? Alright, so now the next slide is something different, because the first two that we just talked about, the lac and trip operons, those are both called negative regulation, negative feedback, but there is a time when there is positive gene regulation. Okay? In addition to repressors, some operons are under the control of activators, things that are actually essentially the opposite of repressors. They can turn up. In other words, make an operon work faster by making it easier for RNA polymerase to bind to DNA, therefore facilitating transcription of the operon genes. So they speed up, basically. Okay, this could also happen with the lac operon, okay, in a certain situation. Okay, I already told you that bacteria prefer glucose. That is their preferable sugar to break down. Okay, so if glucose and lactose are available, the bacteria is going to choose the glucose metabolism. So it's going to break down glucose until the supplies are deleted. Okay, as glucose concentration drops, however, at that point, there's a certain molecule called a cyclic AMP, or cAMP, that increases in production as glucose concentration decreases. Okay, as this increases, the cyclic AMP, okay, this can now bind to an activator protein that's present in the bacteria called a cap. Okay, now the cap concentration actually increases as a result of an increase in cyclic AMP, in cAMP. So what started this? Here's the ultimate starting. Glucose decreases. As that decreases, cyclic AMP increases, causes this cap protein to also increase. Now cyclic AMP can bind to the cap. They stick together. It's an active molecule now, which facilitates the binding of RNA poly to the promoter region. So it enhances the synthesis of enzymes of the lac operon. In other words, it's going to help make the enzymes needed to break down lactose. Why? Because if glucose concentration is decreased, now we have to work on breaking down the lactose. So we want to speed up the production of the lac operon by using an alternative method, which is a positive gene regulation, because we're speeding up the production of this. Okay, now look at this bullet. If glucose concentration is high, this is preferable, and this is actually more often, decrease in the cAMP, so they're not going to produce this cAMP, results in decrease of this cap, which also results in RNA polymerase having a low affinity for the lac operon, so lactose metabolism does not occur. Okay, so it's still working with that lac operon, but it's in a different situation. So here's a picture. Okay, let's look at the picture of it. Okay, here's the regulatory gene. Okay, so this is the regulatory gene. Here's the promoter region. Here's what you are used to seeing as the RNA polymerase. There's the operator, and here are the genes needed to be transcribed. Okay, so in this situation, this is the molecule I was talking about called cAMP. Here's the cap. That's the cap, that certain protein. If this cap is attached up here, like you see in this top picture, if it's attached, that increases the chances of RNA poly being able to transcribe this gene. So in this picture right here that you see, if that cap is attached, this little yellow means it's active. It actively helps RNA poly bind. Okay, it's kind of like giving it a little jump start. So how do we get that? By glucose concentration being decreased, then this molecule is produced in a higher concentration. That's the cAMP. So if this is made in higher concentration, in turn it makes these produced more. So there's more caps, there's more cAMP, there's more binding of the cap to the cAMP. When this binds, it's now active. Okay, look at what made it active. It has a little change of shape right here because that's where RNA poly can stick to. Okay, so that change in shape made that an active cap. Now that goes, transcription occurs, and now the enzymes needed to metabolize lactose are being made. Okay, so that's lactose present, glucose scarce. Look at the bottom diagram. This is lactose present, okay, so both of these have lactose present, but glucose is present, so it's going to be preferred for metabolism. So in that case, okay, check this out, we now have an inactive cap, not going to bind because the concentration of cAMP decreased. If that concentration is decreased, we have fewer of these, they're not activated, they don't come up here and bind, RNA polymerase is less likely. It doesn't mean it can't, because I just showed you a minute ago that it could, but it's less likely to bind, okay, because there's not something that's speeding up the rate of the reaction. All right, let's see. Okay, so the next part we're going to talk about is eukaryotic gene expression in a eukaryotic organism, aka us. Not just us, but this is the most important thing that we like to relate this as talking about human eukaryotic gene expression. And we've actually talked about a lot of these things, so I'm going to go kind of quickly through this stuff because I made sure I included it when we discussed transcription. But there are several ways gene expression can occur in a eukaryote, and we can compare this to a prokaryote. First of all, transcription occurs within the promoter. We've got this catabox. We don't see this in prokaryotic organisms. Okay, and we talked about, if you see in the picture here, the catabox is several sequences of repeated T's, A's, or thymines and adenines. And that catabox is where the transcription factors know to attach to. These transcription factors, when they recognize the catabox, that allows for the binding of RNA polymerase, as you can see in this third picture. So catabox, okay, and the catabox is on the non-template strand. We talked about that. And so then the transcription factors, which are, there's several of them, bind to the DNA, and then RNA polymerase can come in. And we can see in this picture right here where the arrow is, that's where transcription occurs. And the RNA, the new RNA transcript, is made 5' to 3', okay, in that direction according to the template strand. So that's one thing. Okay, the next thing, and we mentioned this as well, is transcription will continue, and we'll keep getting A to U, G to C, A to U, G to C, until somewhere on that strand there will be a polyadenylation, and I can never say this, but polyadenylation signal. The polyadenylation signal is 2 codons. Sometimes they put a little dash right in here where my arrow is. So it's AAU, AAA. Okay, and when that is reached, that signal, the messenger RNA is released 10 to 35 nucleotides downstream from that signal, although transcription continues. Okay, so in order for cleavage and polyadenylation to occur efficiently, mRNA must contain a couple of these they're called poly-A signals, and they're located upstream to the poly-A site, which is where the tail is going to be added. Okay, at this point, RNA is known as the prescript or pre-RNA, and then we're going to do some other stuff to it, which is what we've talked about as well. Here's the same picture we mentioned with the spliceosome. So the mRNA is going to be edited. This does not happen in a prokaryotic organism, because the long sequences of non-coding DNA called introns must be spliced or cut out. So in the top picture you can see here's prescript RNA. Okay, it's already had a 5' cap added to it. Okay, so it's got that little yellow piece. We've got some DNA going, or excuse me, the mRNA here, which is composed of exons and introns. And so in this picture, the spliceosome, which are these things called SNRPs, okay, small nuclear RNA, as well as other protein molecules, come around the RNA, cut out the pieces that are not needed, which are the introns, and we will have a new, right down here, mRNA that consists specifically of exons, which are the pieces that are going to be expressed. So this is eukaryotic gene expression, does not happen in the bacteria. The modification of the cap and tail, we talked about that as well. So the tail is stuck on the end after the polyadenylation signal. See in this diagram you can see there's the polyadenylation signal right there, AAU, AAA. Then the tail is put on, the cap is put on, which is guanine, and then those modifications help in a couple of ways. One way it helps is to help the mRNA leave the nucleus. The other way is to protect the mRNA from degradation of enzymes that are in the cytosol, and also to help the ribosome attach to the mRNA. So there's a couple of reasons that we've discovered that the modification of the cap and the tail happen. Okay, another picture you can see here, we have talked about this as well. This is just the whole picture. Initiation of transcription, how does that happen? Because we have to have transcription factors, we have to have RNA polymerase, and that kind of enhances the initiation of transcription. Okay, elongation, as it gets longer, it's going ADU, 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 G to C, G to C, G to C, G to C, down there. Okay, so ADU, G to C, making this mRNA until finally termination, whenever we get to that polyadenylation signal that's put on there, and then we get our completed mRNA transcript down here at the bottom, which can then be modified by putting a cap and a tail and cutting out the introns. Another picture, just big picture, you can kind of see it all in this one. This actually includes transcription and translation down here at the bottom. So you can see it all in one picture. There's your transcription right there. Here's your prescript mRNA, but you can see in this picture where I'm pointing to with my arrow, it's talking about cutting out the introns so that we have just exons. And then there's your tail, your cap. Okay, here's your functioning mRNA. This is the regular mRNA going out to the ribosome, assembling itself in the ribosome, and going through translation to make our polypeptide chain, aka our protein. Okay? Alright, now talking a little bit more about regulation in eukaryotes. Early in development, eukaryotic cells are what we call totipotent. Okay, I'm going to pause for a second because I need to get my pen so I can write some vocabulary terms on here. Hang on. Okay, got my pen ready. So a couple vocabulary words. Totipotent, which kind of is funny to me whenever you say it, but it's having unlimited capability. So I'm just going to put right here, unlimited capability. It has the capacity to form an entire organism. So cells that are totipotent can actually become an entire new organism. What they say in mammalian embryos is that cells remain totipotent until the 16-cell stage. Well, what do we mean by that? Look down here at the bottom, you can kind of see. The fertilized egg is called the zygote. Okay, so there's your zygotic organism. Divides, becomes 2 cells. Those divide, become 4 cells. Those divide, become 8 cells. Compacted form, and then 16 cell. So until that time right there, these cells are what we call totipotent. They have unlimited capability to be, basically you could extract these and they could become a whole new organism. They're not specialized in any form yet. As far as like time frame, they estimate it's about 4 days after fertilization that it gets to the 16-cell stage. But there's a lot you can do with these totipotent cells because they have unlimited capability. Okay, that cells can be described as pluripotent, which you've probably heard before because we've talked about stem cells. Pluripotent is a little bit different, where they're capable of developing or differentiating into many cell types. Okay, so they can differentiate into different cell types. So we could take, for example, embryonic stem cells. Okay, stem cells from an embryo, and we can harvest them because they are pluripotent, and we can make those cells become whatever cell type we want. Well, what does this mean? Well, you guys already know that cardiac muscle cells and nerve cells are two cell types in the body that theoretically do not divide, or if they do, they don't divide that often. So people who have damage or any kind of accidents or things that happen to their brain, spinal cord, or heart, you know, it's really hard for them to repair that damage. A lot of times it's irreversible. Well, if we can get our hands on some of these pluripotent cells where we can make them differentiate and become a cardiac muscle cell or become a nerve cell, there's a lot of possibilities for what we could do to people who have damage to their heart or brain or spinal cord. Okay, so where do we get most of these from? Like I said, the most pluripotent cells we have do come from embryos, which kind of sets up a huge ethical debate because a lot of people don't want to mess with embryos. You know, it was suspected at first that most of these stem cells were attained from abortion clinics, but that's not the case. Most of the embryos that are used for research purposes today, they actually get them from fertility clinics. Whenever they have fertilized eggs that they don't implant into individuals who are seeking fertility treatment, they can use those to create their stem cells. They can use them for that reason. So like I said, a lot of them come from embryos donated by patients who are undergoing infertility treatment, not necessarily from abortion clinics, which was suspected, you know, years back. People thought that that's the only place you could get them from. And you can also get adult stem cells, but they are not as pluripotent. The idea of this pluripotency is when exposed to certain chemicals in the laboratory, you could take one of these cells and expose it to chemicals that are meant for this cell to become, let's say, a nerve cell. And so you've enhanced the ability and you've now caused that cell to become specialized. Specialized means that that cell has a certain job, a certain function. When it is a nerve cell, it takes on a certain characteristic and function because that's what it's destined to be. All right, so the next slide talks about as development continues with those cells, like I said, they differentiate. Okay, so differentiate means, like in us, differentiation occurs when we start to develop skin cells or epithelial cells, when we start to develop our muscle cells, we start to develop blood cells, we start to develop nerve cells. So differentiation occurs early on in embryonic development, and we'll spend a unit talking about that. But as differentiation occurs, it's kind of cool. They've discovered it's due to, we're tying it into gene expression, what's called differential gene expression, okay, in each cell. So when you look at, let's say, a muscle cell, it's got the same DNA inside of it as a functioning nerve cell. So muscle cells and nerve cells in our body have same DNA, but what makes a muscle cell act different than a nerve cell is because they have different expressions of their genes. So when a muscle cell is supposed to have certain functions, those genes that code for that are turned off when it's in a nerve cell. And likewise, with the nerve cell, genes are turned on with what they're supposed to do, and they're turned off when they're in the muscle cell. So that's known as differential gene expression. That's what causes this differentiation. And so it's different genes that code for different functions in these cells. Now, this is a cool thing down here that I'm sure you've seen before. Some organisms can actually go through a process of look at that D, differentiation, where they make the cells that were already differentiated go backwards and de-differentiate for the purpose of gaining more cells. This happens in starfish regeneration in animals, or planaria, this little dude up here, which we're going to work with in a couple of months. But if you take this planaria and you cut it in half right here, it will actually grow on this bottom segment. It'll grow a new head. Okay, it's kind of cool. You're going to be able to do this in my class. And then this segment down here can grow a new tail. So this is an adult planaria, already differentiated. All the cells that are inside of him they already have a specific job. But because of regeneration in animals, when you cut this planaria, the cells on both ends can actually de-differentiate and grow, in this case, the head, which would grow the cells that do that job. It happens a lot in plants. If you've ever cut a plant before, like here's your growing plant, it's got a root, you can slice the plant in half, plant the top part in water, and it will actually grow its own roots. Take the bottom half, plant it in the ground, and it can actually grow stems. So plants do this all the time. You can be your own cultivator and do that. It's kind of cool. Okay, IPS is a procedure that's relatively new. I think like 2006, 2007 it started coming out. It's induced pluripotent stem cells. It's where they're artificially derived by a human somatic cell, an adult human somatic cell, and you can force these cells to express specific genes. So without having to use the controversial embryonic stem cells, you can force an adult human somatic cell, force it to de-differentiate and by doing that can try to gain cells that now are pluripotent. So this is a big area of research right now that's occurring. Okay, moving on, this gets a little more difficult here. We haven't talked about this before. So in a eukaryotic organism, remember we're still talking about eukaryotes, gene expression is regulated by 3 mechanisms that we're going to talk about in the next couple of slides. Okay, so to regulate gene expression, that means to regulate the transcription, translation of genes, okay, to turn them on and turn them off. It can be done by regulating the chromatin, okay, the actual chromatin in the organism. It can be done by regulating the initiation of transcription, and it can be done by regulating post-transcriptional regulation, so after transcription. So there's 3 main times of regulating gene expression: regulating the actual DNA, regulating the beginning of transcription, regulating after transcription is over with. Okay, so 3 different ways of doing that. So let's get into these. Okay, here's the first slide. This slide shows you regulating of the structure of chromatin. So we're talking about the DNA here. So basically there's 2 to 3 meters of DNA per cell. In our cell, it's elaborately folded. Okay, it's folded around these histones, and we've talked about this before, but here's little histones. These are protein molecules, these little purple things. So the DNA is wrapped, like you can see where my pen is, wrapped around these histones to create a nucleosome. So a nucleosome is double-stranded DNA wrapped around a couple of histones. Okay, it forms a cluster of those. We also talked about how the DNA between these nucleosomes right here is called linker DNA. So what you have is a nucleosome, linker DNA, a nucleosome, linker DNA, nucleosome, linker, all the way down the entire strand of double helix DNA, and you can see where it fits into this picture. Okay, here's a sister chromatid, there's a sister chromatid, or this is a replicated chromosome. So all of this is wound up within this. You can see this arm down here of the chromosome. Okay, so there's stretches of DNA between the nucleosomes called linkers, there's the word. The cluster of histones forms the nucleosome, so that's double-stranded DNA with that histone. Okay, so now, how can we modify this? Okay, here's where you see the folding of DNA is very highly specific. The more condensed the DNA is, the less likely it is to be transcribed. Okay, so what we're going to put right here is it depends on how close the promoter is to the nucleosome. Okay, so as DNA is highly folded, you can see in this picture right here, okay, it's folded back, forth, back, forth, back, forth. The more condensed the DNA is, if it's really, really tightly wound, it's less likely to be transcribed, so it's got to be a little bit looser in order for transcription to occur. And it also depends on how close that promoter is to the nucleosome. Okay, during interphase, DNA is visible, you guys know this, as irregular clumps called chromatin. Okay, so it's less clumped, or excuse me, less whatever, tightly wound during interphase. We have 2 types of DNA, heterochromatin, and down here it's not showing because it's really low, is euchromatin. Okay, we actually discovered that the heterochromatin is tightly packed. And as far as like what it looks like in the nucleus, it stains intensely, it stains really dark. These are the parts of DNA that are not expressed. Okay, so transcription does not occur on the heterochromatin, the part that is tightly packed, most likely because the promoter cannot be accessed, and we can't get the RNA polymerase in there to attach to it because it's so tightly packed. Okay, so those are segments that are not expressed. The euchromatin, less tightly packed, so of course you can imagine there's a huge lag time in my pen. Look at this pen. It's driving me crazy. Look at it. Less tightly packed, therefore actively expressed. So transcription is active on that section, on the euchromatin. Okay, so that's one thing about gene expression as far as the DNA. Okay, the next part is, we also know that these histones, I'm going to draw a little picture for you, don't laugh at my picture. Okay, so let's say here are some histones. I'm going to put 4 of them in here. Here's the DNA wound around it. Well, these histones have little pieces that come off like this. They look like little bad hair days that come out like that. Okay, so they protrude out from the nucleostome. These are called histone tails. Come on, pen. Histone tails. So each little histone has a couple of tails that protrude out from around the nucleosome. Well, that makes them really accessible for chemicals to come in here and modify those tails. So certain chemicals can come in here and interact with that, and if it modifies the tail, that can have an impact on gene expression. So on these tails, there are chemical groups, because these are proteins. You guys know there's chemical R groups that are there. So those R groups can be removed, they can be interacted with by other groups, and that can have an impact on gene expression. So over here you can kind of see in this area, there's a couple things that can happen. Okay, acetyl groups, COCH3s, can be added to the N-terminus end of the histone tail. So on the end of the tail, the N-terminus end, another group can be added. What does that do? It neutralizes the charge. Okay, it's normally positive, but if we're adding on this group that has a negative charge, it neutralizes that. Now this histone is less attracted to the nucleosome, so this doesn't want to stick in there. It loosens the DNA, and now it becomes more transcribable. So we've discovered that in eukaryotic organisms, there is this modification that can occur by taking chemical groups, adding them to the histone tails, and now we've got more transcribable DNA. Okay, kind of cool, right? Okay, the bottom one is even another, a different way of modifying the gene regulation, and that's with DNA methylation. There's a huge discovery in this. This is blowing up right now with research, y'all. I was talking to a friend of mine who's in med school over Christmas break, and they were talking about how there's this field of DNA methylation is just exploding. But this is a way of regulating gene expression by adding methyl groups to certain bases in DNA. Most often they've discovered it happens with cytosine. It deactivates the DNA. So if DNA is deactivated, it's not going to be transcribed. Okay, so the more methylated, the highly methylated that it is, the less transcription occurs at that region. Well, lo and behold, they found out that this is what the reason for the inactivated X chromosome, which we studied with genetics. That in females, remember we talked about one of the X chromosomes is inactivated. We discovered that the one that's inactivated is highly, highly, highly methylated. And so a lot of these methyl groups are added to the DNA, makes them deactivated, and now nothing's going to happen on that as far as transcription. So it's kind of a cool additional way of modifying gene expression. Okay, next slide is talking about transcriptional and translational regulation. Okay, so not in addition to modifying the DNA, transcription and translation can also be regulated. And we've been over a few of these before. A few of these are new, but just different ways that gene expression can be regulated in eukaryotic organisms. Okay, so the first one right here is there's a TATA box. We talked about that. The presence of the TATA box within that non-template strand allows these transcription factors to attach, which means RNA polymerase can bind more to the DNA. So that's one way of regulation. We've been over that. Check. Okay, this is a new idea here that I haven't taught you before, which is these enhancer sequences. So there are, just look at the word, enhancer sequences. They enhance the likelihood of the gene to be read, okay, to be transcribed. So there's certain specific DNA sequences that are located upstream from the promoter region, which binds these little special activator proteins. Well, if activator proteins are bound, that makes those transcription factors bind, which makes RNA polymerase bind. So we've discovered that in addition to the TATA box, okay, which helps the transcription factors bind, that there are certain DNA sequences along the gene. So you look at the gene, and there are sequences upstream of the promoter. So here's the promoter down here. Well, these sequences increase the likelihood that these transcription factors attach, and so that just is another way of increasing the rate of transcription. Okay, we also discovered that in addition to enhancers, we have silencers. So at certain times if the DNA doesn't need to be transcribed, then these bind repressor proteins and turn off gene transcription. And what's even cooler is these two things work hand in hand. They work together to make sure that the rate of transcription in us occurs at a good rate. So sometimes our genes are enhanced, sometimes they're silenced, to make sure that we've got appropriate rate of transcription occurring. All right, let's see. Okay, a different type of RNA, or excuse me, gene expression, RNA regulation. Let me get this on full mode right here. Okay, it's called alternative RNA splicing. And all right, we're good. It's called alternative RNA splicing. Once again, this is a post-transcriptional regulation. Okay, so if you see right here what's listed under, after transcription, we have something called alternative RNA splicing. It's kind of cool when you look at it. So here's your DNA. Okay, here's your exons. Remember, these are the coding regions. These are the good ones. There's, on this one, 5 different exons. So here's your RNA transcript. Okay, something that happens that's kind of cool is you can have different mRNA molecules produced from the same primary transcript. So this primary transcript is going to actually split in half, create 2 functional mRNAs. Here's one. There's 2. They were both created from the same original transcript, but it did a real quick splitting in half. So number 3, which was in this case the important gene that was needed in this mRNA, ended up on this side. Number 4, which was the important gene, ended up in this mRNA. And so those were the important regions that needed to be in those mRNAs. It's kind of a cool thing. So this could actually be, this mRNA right here could be translated into a completely different protein than this one, although they may have related functions. So in our body, this can happen a lot with muscle proteins. We actually have prescript mRNA like this that can be split in half, and we can make a troponin protein of one variety and a different one over here, both used at the muscle, but you know, still come from the same mRNA. It's kind of weird because humans have a surprisingly low number of genes that we actually use. They've compared it to like a soil worm or sea anemone, but out of those, 75 to 100% of those genes probably undergo gene splicing, which means in us, we have a lot of areas that don't code for proteins because we can use the same area and, like this, splice it and make 2 different proteins out of it. So it's called alternative RNA splicing, and it's, like I said, it's a cool field that's also exploding in research. A lot of this stuff is new. These last couple slides, also same thing. We're going to talk about some things that are new that you guys are going to learn a lot when you go to college because it's, like I said, in that field. Okay, this slide has a lot of writing in it, so if you're recording, you might want to push pause and then copy these things down. But I'm going to go ahead and talk. These are still ways of regulating gene expression, but this is post-transcriptional, okay, meaning after transcription. There's several things that can happen that can still help regulate gene expression. What they say is, I say they because I'm not this expert scientist, but basically in prokaryotic organisms, after mRNA is transcribed and it goes to translation, it's broken down very quickly. But in eukaryotic organisms, it can survive for hours, days, weeks, and sometimes can survive longer than that, especially in the case of hemoglobin, and then it can be translated over and over and over again. So the mRNA doesn't necessarily degrade quickly. It can go through translation and be used a second time. And we even talked about the polyribosome effect as well. So bacteria also can do that in the case with these polyribosomes, but most prokaryotic mRNA is broken down pretty quickly. Okay, the second one you see here is translation. Okay, so we can regulate the production of the protein through translation. This occurs at the initiation stage. Well, if for some reason the mRNA doesn't go and attach to the ribosome, then it's not going to be translated. So there are certain proteins that we have, eukaryotes have, where they bind to an untranslated region, but what happens is it prevents the mRNA from attaching to the ribosome. So it's just one more way of slowing down, in this case, gene translation, slowing it down if need be, if we don't need to produce an excess of those proteins. Okay, and the last bit that you see down here is once a protein is produced, or I should say the polypeptide chain, when you get the polypeptide chain, before it becomes a functional protein, it definitely goes through some modification, has to be processed. Sometimes it involves like joining with a second polypeptide chain, or in some cases it'll have like a phosphate group will be added to the end of it, which will help it turn into a different protein. Insulin actually has to be cleaved, so you'll have like this polypeptide chain formed. It actually has to be cut to form insulin. Some have to be transported to their target destination in order to be functioning. So there's a lot of processing that has to occur to the polypeptide chain after it's produced to make sure that it's functional, not just the stuff here that happens in the beginning stages. Okay, this slide is really cool. Wait till you read the stuff in the book. This is a whole new field called MicroRNAs or miRNAs. This is another post-transcriptional regulation. These are special types of RNAs that are non-coding. Okay, it's kind of cool. So genome sequencing, let me just give you the breakdown of this. Genome sequencing has revealed, so out of the last, I would say 10 to 14 years, genome sequencing has revealed that protein coding DNA accounts, so like the coding sections, only accounts for about 1.5% of the genome. So when you look at our DNA, only about 1.5% of our DNA codes for actual proteins. That's bizarre. We're saying only 1.5% of your DNA codes for a protein. What does the other part code for? Big question mark, because for years we thought that's what most of the DNA coded for. Okay, so for many years scientists thought there was a lot of untranscribed. They thought, oh, it was just extra fluff DNA. Research is suggesting that a significant amount of the genome is actually transcribed, not into proteins, but into non-coding, okay, non-coding RNAs. So this is kind of weird. So we think that most of our DNA, as opposed to coding for proteins, most of our DNA is coding for non-coding RNAs, meaning not messenger RNA, not tRNA, not ribosomal RNA. Our DNA is actually making these non-coding RNAs, okay, called other RNAs. Here's one of them. This is the hottest one right now. Actually, these are both really, really hot in the research field right now. This one is called MicroRNA. Okay, short is miRNA. It's made from a longer RNA strand. Okay, so you've got like a long RNA strand that folds onto itself to create a hairpin loop. It almost looks like tRNA, but it's got many, many more folds. So it goes like this, like this. It's got many, many folds to it. Okay, so it folds upon itself. It works with an enzyme on this RNA called a dicer, which I kind of like, sounds like slice and dice, which is what it does. It trims it to a short double-stranded fragment. Okay, the job of this, though, look what it is down here. Here's the functioning. This whole microRNAs, what they should do is block translation. So they're going to turn the translation of the protein off. Okay, so as one strand is degraded, the remaining strand can bind to any complementary mRNA and turn it off. I'm going to show you in the picture in just a minute. Okay, so this is kind of a cool thing. Okay, there's also another field exploding right now called siRNAs, small interfering RNAs, similar to these. Okay, original mRNA strand is longer, but there's more hairpins, so they're more highly convoluted than these. Generates many more siRNAs. Okay, and like I said, there's not a lot known about this bottom group. They just know that these exist, and now they're doing more research to try to discover those. So let me show you this picture. It's cool. All right, so here's the miRNA. So what you have is, here's the miRNA transcript. Okay, so this folds back. See the hair they call it hairpin because it looks like a bobby pin. You guys that wear bobby pins in your hair, you girls, they look like that. So this is a hairpin loop. Okay, loop, 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 all around like this. So the little orange fragment that you see there, that's the miRNA right there. So it folds upon itself, but there's the miRNA. Okay, so it's hydrogen bonding with other sequences like A to U, G to C, A to U, G to C. Okay, so what's going to happen, I'm going to take you through a little series here. This is step number 1, okay, in this picture. So there's an enzyme that comes in that's going to cut here and there. Okay, this is not the dicer. This is just another enzyme. So an enzyme comes in, cuts the hairpin from the miRNA transcript. So it's going to cut here and here. Number 2, the dicer, which is another enzyme, looks like Pac-Man, you can see it right here, is going to trim the loop. Okay, so it trims the loop and the single-stranded ends from the hairpin. So it's going to trim it, cut those off, trim it, cut that loop off after the other enzymes up here cut the hairpin. Okay, so this has to be taken away from the rest of this. Okay, so number 1, enzyme cuts this away, separates it out. Enzyme number 2, the dicer, trims off the loop, trims off the end that was right here. And now we're left with this right here, this little segment. Okay, so number 3, one strand of the double-stranded RNA is degraded. Okay, so this side over here is going to be degraded, broken down. Okay, the other strand, now we have the miRNA isolated right here, forms a complex with proteins. So here's other proteins, that little purple thing. So we've got an miRNA, we've got proteins. Okay, now the cool stuff begins. It binds to any target messenger RNA. So down in this picture, look at this. There's messenger RNA. The job of this miRNA, okay, 2 things can occur. 1, it can be degraded, so this miRNA chops up the messenger RNA and degrades it, or it can attach, okay, leave areas where they're not attached, you can see it in there and here, so it binds loosely to that and prevents the ribosome from being able to go down the mRNA and translate it. So the whole function, if you go back, the whole function of these is to block translation. So it's one more way of regulating gene expression because we can block it by using these small microRNAs. Kind of cool, huh? Okay, we're going to stop there. That's it for this lecture video. The next one is going to be on cancer. So this gives you enough to work with before I post the next one of cancer, and I'll let you guys know when I do that. Okay? Have a great day, y'all.